Good evening, everybody. Welcome to the third Friday evening lecture of our 2022 season of this wonderful tradition that is the MBL's Friday evening lecture series. I have just a few things to say. First of all, my name's Sebastian Lay. I'm MBL's Chief Advancement Officer. That has no bearing on what I'm about to tell you. My duty tonight is to please ask you to be polite, to silence your cell phones. We're in for a great talk tonight, so we don't want anything to interrupt that. This lecture is being recorded and simulcast on Zoom and YouTube, so just for your awareness. And finally, there will be time for a Q&A at the end of tonight's talk, and then inevitably there are more questions than time, and we do follow the lecture with a reception over on the back terrace of Swope. So you can join our speaker this evening over there after the lecture. And then it's really my pleasure this evening to welcome a tremendous colleague who has joined us just in the last year, Anne Sylvester, who leads our Department of Research. And she joined us from the National Science Foundation and before that, the University of Wyoming. And she's been a tremendous addition to the team. And we've really kind of thrown her into the deep end this summer into really our first full force summer back since COVID. And so I hope you'll welcome her as she welcomes tonight's speaker. Thank you.
Thank you for that welcome, Sebastian. I think I'm learning to swim, so thank you for throwing me into the water. I am very pleased to be here tonight to welcome all of you to our Friday evening lecture. I'm especially pleased to introduce our speaker, Dr. Mandë Holford. I first met Mandë, I think we were both new to MBL when we met each other, and I've learned an enormous amount from her already about what you're going to hear tonight. She is an associate professor in the Department of Chemistry and Biochemistry at Hunter College at the University of New York. She also holds scientific appointments at American Museum of Natural History and Weill Cornell Medicine. And I'm telling you that because it shows you the breadth of her knowledge and experience, ranging from chemistry all the way through natural history to medicine. And we're going to hear about that, how mollusks to medicine work. Some of you may be familiar with the organism she works on, cone snails. These mollusks are beautifully patterned shells, but that beauty is deceptive because they're also extremely toxic. They produce a venom that is deadly to humans and other animals. And so there are many questions around that Mandë has been addressing. How did they evolve? How are they developing? What is this toxin doing? How can it be protecting the animal and other animals? How do they survive it? So she takes a very interdisciplinary approach to answering these questions. From chemistry, she's studying evolution, development, physiology. But I think what's really amazing is that through her scientific breakthroughs, she takes this a step further to think about impact of the research. She asks questions like, how can we harness this venom for therapeutics, for medical advances? Once we understand it, then we can know what to do with it. So she has a great interest in natural products from marine organisms. So we're going to hear about this breadth tonight. And furthermore, we're going to hear about her engagement that is very far-reaching in science. She's a globally engaged collaborative scientist. And she's also interested in science diplomacy. I hope we'll hear something about that. So for tonight's lecture, Killer Snails, Agents of Change and Innovation, let's welcome Mandë Holford.
Hi, everyone. Thank you, Anne, for the really nice introduction. And thank you to Anne and NEPOM for the invite to share my work. I'm really honored as a newbie to MBL to be a part of this Friday evening lecture series. I brought with me 3 of my grad students, and they're also newbies, and we're We came here last week all bright-eyed, and we're still, you know, the eyes are getting smaller, but still pretty big. And tonight, what I would hope to share with you is some of what Anne was saying, how a peptide chemist who was trained at looking at, you know, white liquids and white tubes got involved in wanting to know more about the molecules and how the molecules actually are evolving in the organisms themselves. So I go from peptide chemistry to organismal biology, and that's sort of the story that I hope to share with you tonight. And the organisms I work with are these beautiful shells, which are snails. These are the dead shells, but they're killer snails, and agents of change and innovation is really what I think they are and how nature is showing them. So I hope I can convince you of that. And so when many of you think of venomous animals, right, this could be a loud evening. It's a Friday night. I'm from Brooklyn. I can deal. And so when you think of venomous animals, what do you think of? Mostly snakes, spiders, scorpions, snails, maybe all of the above, none of the above. And so when many people think of venom and venomous animals, they think of them mostly as agents of fear, which makes sense because a lot of them are lethal and can kill you. But when I think of them as agents of change and innovation. And that's because how nature has thought of them. So I'll go back. We went forward a little bit too fast, yeah. That's how nature has thought of them. And so here it is. This is a snail, a cone snail under the sand. The orange thing is a proboscis. On the tip, it has a harpoon, which we call a radula, filled with venom, and it's going to, you know, well, you'll see. Yeah. And this is real time. We did not speed this up. And this is why I no longer just work with clear liquids and white tubes anymore. So the snail is going to come out of the sand and swallow this fish whole. So if this is an innovation in nature, I don't know what is, right? And so here you have a slow-moving, not even moving snail, able to feed on something that's much faster, more agile. And why is this possible? So you'll see this again. This is Conus bullatus, the first snail that I've worked on. And it's going to envenomate, that's what the technical term, it's going to envenomate the fish. And again, the fish instantly goes paralyzed, it's stiff, and the snail will engulf it whole into its rostrum. And so this is because the venom that these snails are using, similar to snakes and scorpions and spiders, it's very fast acting, it's very potent, and it's very specific. But it's also dose dependent. So you'll see that this fish didn't get a whole lot in the first shot. So it's going to wiggle about, but it's just going to get another barb of venom, and that's it. Goodbye. No coming out of the mouth once you get in. And so what the venom is doing is basically it's taking what usually is a physical warfare and turning it into a biochemical warfare, in which this clicker is really sensitive. In which I have to stand right here. In which the components of your venom arsenal is what's going to dictate who the victor is going to be, right? And so this pattern that we saw with the snail has been repeated over and over again in nature. And so this is a phylogenetic tree, kind of like a family tree, how you're related to your mother, your father, your sister, you know, 23andMe. And everywhere you see red is a venomous organism. So again, most people think of snakes, spiders, and scorpions. Not many people think about platypus, or butterflies, or larvae of moths, or, you know, starfish, or octopus and squid. There are a lot of cephalopods people here. You know that as well as I do. And so we think that maybe 15 to 30% of animal biodiversity is venomous. You can find them in the land, in sea, and in the air. And so they're very ubiquitous. And people don't realize that venom is everywhere. It's omnipresent. And so this pattern that nature has repeated, I think there are three main reasons why it's worked so efficiently, right? So the power of venom is actually threefold. The first is that venoms are complex mixtures. They're not just one thing, but they're several things. I like to call it a cluster bomb, actually. And so in the venom, you have both small molecules, peptides, and proteins, where peptides are just smaller proteins, less than 100 amino acids. And so in my lab, we focus mostly on the peptides. So I did, as I said, my training in peptide chemistry. And as a kid, I grew up in New York, and I wanted to go to the museum and love the museum. I wanted to be a museum scientist, but they don't hire chemists, right? So when I saw that video, I was like, here's how I get to a museum, because the venom is made up largely of peptides, which I know something about, right? So each snail makes anywhere of up to and more than 250 unique peptides in its arsenal. It's a lot of peptides. And the thing that's really special is that no two venom arsenals are exactly the same. Whether we're talking about two snails or snails and snakes, venoms are really species specific. So each cocktail really is a unique arsenal of biochemical, cornucopia for us to discover. And so the second thing about venoms is that even though the arsenals are quite different, they all target very conserved physiological things. And so the big three that I like to highlight are blood, brains, and membranes, right? Because these speak to the integrity of the organism, right? And so when they attack blood, they're hemotoxic. When they're attacking the brain, they're neurotoxic. And when they're attacking membranes, they're cytotoxic. And so a lot of the early work on these peptides from snails in particular were done where they were being used as neurotoxins. And so when we say that they're attacking the brain or attacking neurons, what we mean is something like this, where here you see one neuron signaling to another in a synaptic junction, and the peptides in particular from these snails, what they do is they sort of manipulate how those gates on the membranes of the neurons, which are called ion channels and receptors, it manipulates how they work. And so I mostly focus on the snails, right? And so I'll talk to you about the snails, but each organism that's venomous has these kinds of neurotoxins in its arsenal. And that's the third reason that it's so powerful. It's because it works. We know that it works. You just saw a snail eat a fish. So you know that it works, right? The challenge for scientists is figuring out how does it work? And how does it work so well at manipulating cellular communication? And so that's sort of what I'm trying to do in my lab. So we study venom evolution using a comparative model, comparing mostly snails to other organisms. We then try to identify new peptides in the venom arsenal, and then characterize how they function at the gates or the membranes of cells. With the long-term goal of hopefully trying to identify an application for some of the peptides in pain and cancer, which is really trying to find an application for how these peptides manipulate malfunctioning signals in pain and cancer. And so I'm going to start with a little bit about how we discover these venom peptides. So the peptides come in 3 different flavors, sort of, and not the peptides. The snails that we work on are from the Conoidean superfamily, and they come in mostly 3 flavors. So cones, and a lot of the work from cone snails came out of the lab I did my postdoc with Toto Oliveira, and they produce conotoxins. Then you have terebrids, which produce terotoxins, and turrids, which produce turotoxins. And so because in order to have my name on the door, I can't do exactly what Toto did, we focus mostly on terebrids in my group. And the way that we discover them is a little different from what was done traditionally. So traditionally, size and quantity drove which snail venoms were characterized because of the techniques that you needed to use. They required large quantities of venom. And so when I started, the techniques had advanced, right? So we had a lot more omic techniques that required less samples. So essentially, when you used to go out to the field and you needed to collect 80 to 100 of the same species, now we really only needed about 5 in order to get the kind of information that you used to be able to get before. And so what this has enabled us to do is to use evolution and diversity as sort of our guiding light to understand how we interpret the relationship of venom arsenals and how we can do sort of more target-driven, ask more target-driven questions about how the snails relate to each other and how their venoms then evolved over time to have these different peptides in them. So to do this evolution and diversity strategy, we have to start by building the 23andMe. So we have to start by building this family tree of snails. And then once we have the phylogenetic information, we then compare it with the venom information itself, what's in the arsenals. And so that's where these omic techniques come in. So we combine transcriptomics, which is basically tells you, you use RNA to tell you all the peptides that have the potential to be expressed in the venom arsenal, with proteomics, which is the crude extract from the venom itself. So when you put those two together, you get what's called a validated database of what is actually being produced by the organism. And you can then go on and study the peptides for us, and proteins and other things, but mostly the peptides for us that's there. So I want to give you just a little bit on the venom anatomy. So In order to do those kind of phylogenetic and omic work, you need parts. So you need parts of the animal in order to do that. To build the phylogeny, we mostly use samples from the foot to get some DNA. To do the transcriptome work and the proteomic work, we use the venom duct, RNA for the transcriptome and the actual, as I said, extracts from the duct. And then the rest of it is just a nice case. And so all cone snails have this venom apparatus, right, where they have a venom bulb. It looks kind of like a kidney, pumps the venom through the venom duct into the radula sac, where these radulas are lined up sort of machine gun, and they can fire them off. They fill them up and then fire them out through the proboscis. And so when you dissect, what you see is like a spaghetti. Basically, all the fuzzy spaghetti is what the venom gland looks like. It's very long, and it wraps around kind of like intestines. And then the radulas look like this. They're very thin, very narrow, and they're barbed, and that's why we call them harpoons, because they can actually tether to the prey and make sure it doesn't get away. So all cone snails have this anatomy, right? But not all terebrids do. And when we started doing this work, all we knew about terebrid physiology was from a 1970s studies by Miller, in which he described 3 different physiologies for the terebrids. And so there was a type 1, which did not have a venom apparatus. There's a type 2, which did, and a type 3, which did not. So we were mostly interested in trying to find the terebrid samples that had the type 2 venom apparatus. So meaning similar to cone snails, they had the venom bulb, they had the venom duct, the proboscis, and all of the other features. And they were actively using venom to subdue their prey. And so we built the first phylogeny for this group, in which we sort of mapped the presence of the Miller type to the location of the species in the tree, right? So the clades, basically. And so starting from the top, we found that the sister group had a venom apparatus, and then the next one, clade B, Oxymeris, did not. Clade C had the venom apparatus. Clade D had the venom apparatus, type 2. And then clade E, at the time a junkyard clade, appeared not to have it. So by building this phylogenetic tree, we basically developed a roadmap for identifying all the lineages of terebrids that were actively using venom to subdue their prey, so that when we go out in the field and to collect, we can focus on those clades and on those specimens specifically to help us do our work. Because these snails are not huge, and when you dissect them out, the glands are very tiny. And the worst thing is after diving or scuba or snorkeling all day to get back to the makeshift lab in the field to discover that you don't have any venom glands when you dissect it out. So it's kind of like, going hunting for rabbits. When you find the ones you want, you get very smitten and you're happy. And so we're like, yes, we have a roadmap to help us figure this out. And so to expand on where we build out our tree, there are about 400 species of terebrids. And so in the 10 years that we first started to now, we've gotten about half of those species onto our phylogenetic tree. And so we just recently filled, so everywhere you see a drop is where we've conducted expeditions with the Paris Museum of Natural History to identify these species. And so we recently published a revised phylogeny where we dated the terebrids to about 50 million years ago. And again, those clades that had the venom apparatus continue to have lineages that all have the venom apparatus present. And then the junkyard clade, that Myarella clade, we've now identified also has a few lineages that have the venom apparatus. So there's the McLauer clade, the Profunda terebra, and the Punctu terebra clade. So what I really, really love about studying these terebrids is because unlike cones, where, again, all of them have the venom apparatus, terebrids didn't. And so they've lost it at least 8 times throughout their evolution. So they're really a model system for how you can study how venom evolved in this group. And we know the ancestor had a venom apparatus, and then it's since been lost. And so the diversity that you see here, especially in these new genres that we've identified in the Profundi terebra and the Neoterebra, is really astounding because they're found both in deep water and in shallow water. They're found in the Indo-Pacific and the Atlantic. They're found with a radula, without a radula. And so there's a whole big, very diverse, when we did the morphological analysis of this group of the foregut anatomy, we found that there are 12 different types of anatomies for this group. And so those 12 are, the first 3 are not the same as the Miller 3. These are our unique types, but they're found all throughout the tree. And then again, for us, the important parts are the ones that are actively using the venom gland to find their prey. But the vaginal tube that they're using is very, very varied, right? It's very diverse. So within the Terebrids itself, you find all the vaginal types that are found within the Conoidean families. So again, making it a great model for studying how these things have evolved the ability to prey or defend themselves using venom. And so something to know about how venom is metered. The morphological constraints on the venom really depends on the anatomy of the animals. So like sea anemones, they just release the venom. It's not metered at all. But more complex organisms like our cone snails and terebrids, they have direct venom modulation. So to the point where in the venom bulb, they can pump, if they need to do, if they're feeling threatened, they will eject defensive venom, which is proximal to the venom gland. And if they are on the hunt, they'll eject predative venom, which is distal to the venom gland. But it's all one duct, right? So why aren't they mixing together? Why don't you get a mix of every kind of venom all the time? Very interesting question. And it's been shown also, again, in the assassin bug. Similarly, in its glands, the posterior glands are for predation and the anterior glands are for defense. So these is a very complicated foregut anatomy for manipulating not only what kinds of venoms are being expressed, but how they're being used as well. So to give you a little bit more about the peptides, these peptides are expressed as a single gene, and so they have a signal sequence and a pro-region that's conserved, and then on the end, there's a cysteine-rich mature peptide which gets cleaved off, and that's the toxin, that's the bioactive material. But this is widely hypervariable apart from the cysteine frameworks. So you can think of it kind of like a stone and a chameleon. So on one end, nothing's changing, which is good because that's sort of the constant thing that we can use to trace the evolution of these things throughout time. And on the other thing, it's always changing. It's mutating in what's like the arms race between the predator and the prey. And that's what you find here on this end of the peptide, right? And so using transcriptome libraries, we're able to identify the gene superfamilies, the stones, that are being pretty constant. We use the signal sequence to group our gene sequences into these families. And what we find is the families are usually linked to a specific cysteine framework. Not all the time, but most of the time, right? So like, for example, the L superfamily has 4 cysteines, but they're in a different framework from the J superfamily. And this is a consistent way for us to group the peptides and then also help us predict potential function, because the cysteine framework is like a molecular glue. It holds the peptide together, so they behave more like microproteins, and they don't get degraded as quickly in the system. And so at the bottom, I'm showing you 2 structures from the gene superfamily to the cysteine function of framework. We got the M7A, which is a peptide that's on the market called Prialt, that's used to treat chronic pain and HIV, and TV1, which is a peptide we've identified that seems to have activity in liver cancer. And so both of these have very distinct folds that their cysteine scaffolds help them to then determine what's going to be potentially their molecular target. And so we've identified 27 new Terebrid gene superfamilies, 27 new stones that are very different from the conotoxin superfamilies. There's only one that's identical. And we've created a database of over 3,000 new novel Terebrid peptides. Again, very different from anything that's been found by, in cone snails, which is why I have the Holford lab and not the Oliveira lab. And so now I want to tell you, what are we going to do with all of these peptides, right? So the diversity of the venom peptides is kind of an important thing to try to dive into. And so we did some killer snail venom evolution mathematics. So venom is a molecular trait, right? And so the thinking is that if we're able to then correlate the venom composition, genotype, like what's in the venom, to the prey, what the venom is being used for, we can then trace the adaptive evolution and ecological processes of the Terebrid snails. So basically, it's like, how does the diet influence the composition of the venomous animals? And so there are 2 prevailing theories. One is that the prey preference can determine venom components. Basically, you are what you eat. The type of compounds in your venom is dependent on what you're eating. The other is that there's a positive correlation between the dietary breath and venom complexity. So the numbers of compounds in your venom is dependent on if you're eating on very simple things like worms or very complex things like fish and other things, right? So the question we asked does repeated innovations in the Terebrid foregut anatomy and venom composition correspond to diet variation? And to do this, we basically looked at the gut meal of 17 species of terebrids. We had 71 specimens, and these terebrids mostly feed on worms. And so what we found is that terebrids eat a lot of different kinds of worms. We found 18 different types of annelids in the guts of these terebrids. And what's interesting is the ones that are highlighted red in the tree are all the ones that have a venom apparatus. So even the ones that did not have a venom apparatus are still feeding on worms. And so how are they doing that? So there could be that they're releasing the venom into the water to make the worm go quiescent, like we've seen with cone snails. They can do that, and then they'll engulf it. Could be they're using the accessory feeding organ, or their peptides are being expressed in salivary glands, but not sure how they're delivering it. There's a whole lot of questions that came about just from this that we still have to dig into. What we also found is that terebrids without a venom gland have a larger diversity in their diet than those with a venom gland. And this is similar to what was found with snakes. So snakes that have venom glands feed on usually the same things versus the ones without, which kind of roam around and try to find whatever they can find to eat. And then really interesting was that terebrids with different diets seem to be producing much of the same kinds of peptides. So this is what we call a heat map. Here on your right are different species of terebrids. On the left are the mealworms that they're feeding on, and across the top are the cysteine frameworks, so in Roman numerals. And so you see, for most part, these all are feeding on different worm meals, but their terebrid arsenal, venom arsenal, is exactly the same. An exception is the Hastula hectica at the top, which seems to have a very unique worm meal and also a very unique venom arsenal. And it could be because this one is really found at the intertidal, so it's right at the shoreline. And so what's available and what's happening there is quite different from the others. What was surprising to us was we did not find a direct correlation between venom diet and venom complexity. So whether they fed on neanthes or fireworms or Scoliolopsis, the toxins were all going to be the same. And by complexity, what we mean is either the number of putative venom peptides or the number of cysteine frameworks. This was quite different from what was found in snails, in cone snails. In cone snails, there seemed to be a direct correlation in the number of putative toxins and the number of cysteine frameworks. And so one reason we think this is different is, again, looking at that crazy anatomy chart. Terebrids really are sort of a microcosm of different kinds of evolutions. And so that might be why there's not a direct correlation between what you eat and your diet. But that's just a guess. And again, we used 17 specimens, so we have to repeat this. So where are we going with this evolution of terebrid story? In the next steps, we really want to figure out how have terebrid venoms influenced the global patterns of biodiversity, and does evolution of the Terebrid venoms follow other arms races like microbial, host parasites, and plant pollinators? For the last part of the talk, I want to talk to you now about the function of these venom peptides. For the most part, venoms have been used to kill. They're lethal, right? So there's this evil Captain Venom who can envenomate you. And similar with our snails, certain species of these snails are very lethal, mostly in Australia, and so don't go anywhere there. But so when people think, how can venom become a drug? It's because what we're doing isn't injecting the whole venom arsenal into you. What we're looking for are those individual peptides that are very good at manipulating signals, especially malfunctioning signals, and figuring out how we can use that for good, right? So Dr. Envenema is here to identify those peptides in the venom. And it's happened successfully. So there have been successful venom compounds or peptides that have been derived from venom that have been turned into drugs, right? The first one was captopril, which is used for hypertension for the most part, right? And then you have others as well. angiomax, which is deep vein thrombosis. You have exenatide, which is used as a diabetes drug to treat type 2. And then the breakthrough drugs from cone snails. The first and currently only drug on the market from a killer snail that's been used to treat chronic pain in HIV and cancer patients. And what's amazing about this drug is that, unlike morphine, it doesn't work on opioid receptors, so it's not addictive. It does have a very serious drawback in that it's not peripherally active, so you have to get a spinal tap to deliver it. So it only is being used for people for whom morphine is no longer an option, or they're in really serious pain. But what's exciting about this chart, or this diagram, is that not only are these peptides showing us new compounds for treating diseases and disorders, but they're giving us new mechanisms and new pathways for doing that. Prior to the discovery of Prialt, most pharmaceutical companies were looking for opioid-acting components to treat pain. Now they know that they can go elsewhere, and then that, of course, minimizes the addiction, the opioid addiction that we're all going through right now in the country. So when I started to think about applications for these, I wanted to think about pain as one of those. And because the terebrid toxins are so different from conotoxins, we wanted to see if we can find peripherally active analgesic compounds. And so we did that basically by looking, again, using the transcriptome, looking at cysteine frameworks, identifying those that seem to be peripherally active, using whole animal models to then test if we can either feed them to flies or inject them peripherally. Do we then see a response in a heat avoidance assay, which would indicate that there's some analgesic activity. I won't talk more about that because it's been published. And then the other area that we're looking at is a cancer. So thinking of cancer as a channelopathy, the gates on the outside of those cells. So you have a tumor cell in pink, the healthy cell in green, and the idea is that we use the selectivity of the peptides to really target overexpressed channels in tumor cells so we can make more selective cancer therapies. Because one of the big things about cancer treatments is that it's not discriminate, right? So it kills both tumor and healthy cells at the same time. But with these peptides, because they're so specific for certain channels, we might be able to find selective treatments that way. And so this idea has been sort of shown in other things. So from the Western honeybee, there's melatonin, which is 26 amino acids long. It's been indicated in several different types of cancer, and it basically works by activation of apoptosis, inhibition of metastasis, or inhibition of epithelium mescalyl transfer. Then from the scorpion, we have RK1. It's 14 amino acids long. It's been indicated in glioma, glioblastoma, and melanoma, and it works by suppressing metastasis and angiogenesis. And then the last one I'll share is the Chinese cobra, which has done CTX3. It's been indicated in breast cancer, and it works by suppressing EMT. So basically, again, not only are the peptides being used as a potential treatments for cancer, it's the ways in which they're being applied that's so diverse that allows us very different pathways into finding novel treatments. So whether it's using programmed cell death, or you're going through the metastasis, the spread to different sites, preventing blood vessels from forming or preventing the cancer from becoming migratory, all of these are legit pathways that pharmaceutical companies are trying to investigate, and you can find that from peptides in the venom. So it's not such a really rosy story though, right? So the history of peptide drugs has a bumpy road, one of which is delivery. So about 100 years ago, the first peptide drug, insulin, was discovered. And for the most part, we still have to inject insulin because it's not orally active, and the predominant way is the same. And then a major breakthrough happened around the late 60s, early 60s, when Bruce Merrifield invented solid phase peptide synthesis. Bruce was at Rockefeller when I started my PhD program, my PhD at Rockefeller, so I got to meet him. And it was really a breakthrough in the field to be able to synthesize these peptides synthetically. Similarly, we're hoping that Venomics will become a similar kind of breakthrough. And like I mentioned before, there are already venom-derived peptide drugs in the market. And because of the efficiency with which we can mine the transcriptomes and proteomes of these and metabolomes of these animals using Venomics, we're hoping that we can sort of lead to breakthroughs in drug discovery that way. But going back to our killer snail evolution mathematics, we know again that yes, venom is a molecular trait, and in order to manipulate the venom arsenal, we need to be able to manipulate the genes. So speaking as a chemist, a lot of the people who've been working on these venoms are folks like myself, natural product chemists, who focus mostly on the peptides. So it's kind of like focusing on the car, right? But in order to really understand why the peptides target the channels that they do and how they function that they do, we need to figure out how the peptides are being expressed and how they're being regulated. So we kind of have to go back into the factory, to the assembly line, and understand that. And so that brings me to why I'm here at MBL, right? So there's no model system for studying venom, and a lot of what we know about genes and genetics and peptides and how they work comes from studying these great model systems that have been developed. And so the current thinking, this is Conus tulupa, go back one, and then it'll show. So it feeds by engulfing a school of fish, and then it will envenomate them each, and then have itself a meal. So if we want to understand how these genes became weaponized, we think that most of the venom genes were co-opted from housekeeping genes and then duplicated and neo-functionalized, like got shiny, basically. And so if we want to understand how these genes got weaponized, we have to have a system for manipulating those genes and figuring out how we got this ecological advantage to occur and potentially translate that to therapeutic advantage. And so developing a model system for these led me to think about organoids. This clicker is so sensitive. So organoids are 3D representations of organs that are, it's basically, evolution in a petri dish. And so what we were thinking is, can we make venom gland organoids? And this happened mostly during the pandemic shutdown because we weren't able to go out and collect and get new species. So these are tropical marine environments. They're not found in New York, and you can't breed them in the lab. So you can't breed them, and you have to sacrifice them to get to the glands. So how can we build our own sort of model and factory? And so the idea was, can we stockpile these venom glands like we did beans and toilet paper during the shutdown, so that we're able to have them whenever we want them in order to work on them. And importantly, we can have a way of figuring out how the venom peptides genetically are manipulated and regulated. And so we started, most of the organoids that have been made, or I should say all of them, are made from epithelial cells and from vertebrate systems. So this would be the first invertebrate organoid if we were able to do it. And so what we started with was looking at the growth factors. Most organoids are made from the Wnt systems. That was developed in Hans Cleaver's group. And so we looked for those kinds of growth factors. And we were very happily surprised to find that they were present in our snails, and we tested a few worms as well. And so this was like, all right, if the growth factors are here, let's try to see what's possible. So far, we've been able to successfully culture a few glands and keep them growing for a few weeks. So this, considering that there's no system, we were very happy to see this, right? And so we saw active cilia, that's what's shown here in this movie. And then when we stained with collaborators, we stained for DAPI, which is in blue, that tells you it's the nucleus. We stained with the mitotracker, looking for organelles in the cell, and then calcin live, which is for the modal cilia. So all of this made us very, very happy that maybe this is possible, right? So what we're thinking is that if we can combine organoids with venomics, we can really help to advance venom drug discovery. And so that brings me to MBL, right? So this year I'm a Whitman fellow and working with my group, my 3 grad students in Roe, so come by and see us. And we're very excited to be here and experience this, because this is where model systems from marine organisms are made, right? And so there's one cell line for invertebrate snails, and it's the BGE cell line. It's from a freshwater snail, and it was developed in the 70s. So it is possible. So going on the hope of this one cell line, I arrived here last week, and the MBL fashion, you start to talk to people. And I was speaking with Josh and telling him this great story of why we're here and what we're hoping to do. And I got the raised eyebrow emoji. It's like from someone who knows a lot more about this than I do. And he's like, yeah, okay, maybe. I hope you can do it. And then he immediately connected me to his postdoc Juan, who's amazing and has also been trying to make an invertebrate cell line. And he collected me to Carrie. And Carrie started telling me about her work with squids and that the squid heart, once you remove it, can live for a couple of months. So we might not even need organoids, right? We can just maybe do, not the squid heart, the squid ink sac where they deliver their venom can continue to pump and make ink even after outside of the animal. So I was like, now I have the happy face emoji because it's possible that we might be able to get this done, again, through collaborative work here at the MBL. And so another project that we're hoping to start is one on these vampire snails. These are snails that are actively sucking the blood. They don't kill the fish, right? They go in under the gill, and then they're actively sucking the blood of these fish. This is a project I'm doing in collaboration with a colleague, Maha, from Harvard. And again, it's following the MBL tradition. So when we got here, Lewis was telling me, that there's another group that used to work on cone snails from a long time ago, and I met them at dinner, Bruce and Barbara, and they were focused on the function of coagulation and platelet proteins, and they had a lab here for 17 years working on cone snails. So again, I feel like we're in the right place to make this happen. And I'd just like to end by saying I'm not just a Whitman Fellow, but I'm an E.E. Jest Fellow, and that's a really humbling honor for me because I think he's an incredible scientist. And I wanted to end with this really nice quote that he has where he says, "We feel the beauty of nature because we are part of nature and because we know that however much in our separate domains we extract from the unity of nature, there's unity remains. And although we may deal with particulars, we turn finally to the whole pattern woven out of these." And so to me, that reads like my story in the person who started just with the peptides of a white liquids and white tubes. And then we get back to nature, to the snails, and that's how we're going from mollusks to medicine. And so I think that the field of venom, of course, has a lot of potential. This is for all the grad students and postdocs in the field, and some scientists who might be thinking what to do next. Come to the venom side, put on your venom boosters, and help us to figure out these problems, because it's very interdisciplinary. We need chemists, we need biologists, we need evolutionary biologists, we need organismal biologists, we need people who want to make new models in order to really make this lift off. And so I hope that I've convinced you that venom and venomous animals are not just agents of fear, but they're actually agents of change and innovation. And with that, I'll thank all the lab members, past and present, because we were talking about some old stuff and some new stuff. And I'll highlight my 3 lab grad students who are with me, Pravina, James, and Favor, if they're in the audience somewhere. And so please reach out to them, as many of you already have. And then, of course, this is collaborative work, so all of the collaborators that we've done this with and the funding agencies that have supported our work. And I'll end with saying happy birthday to Laura, who's been like our MBL ambassador, and she's taught us where everything is and where to go and how things are going. So happy birthday, today's her birthday. And thank you, everyone, for your attention.
Director's privilege.
Great talk, Mandë. So I have a question. So, there's the well-known story of the bark scorpion, and then there's a mouse called the grasshopper mouse that's not just immune to the venom, but actually it's an analgesic. But are there animals that are completely immune to the venoms and the snails you work with, or are there so many peptides that that's just not possible?
And there are a few, like the mongoose and the cobra, and there are a few in which you actively can see this arms race happening, right? Where there's a race between the venom trying to subdue the animal and then the animal fighting it off. But because of the sheer numbers that are in the arsenal, it's always this ongoing battle where the venomous creature, for the most part, will win and can win and lives to subdue the animal at some point. Yeah, it just gets overwhelming.
Hi, Mandë. When you see the snail species that lose the venom and the venom apparatus, what level is that happening? Is it happening at the genomic level where they just actually also delete the genes, or is it more of a regulatory?
Right, so that's a good question. We don't have a genome for the terebrids. Just last year, there was 2 genomes solved for cone snails, and it's the first ones ever. So we haven't figured out exactly if it's, because we know the ancestor had a venom apparatus, so the assumption is that it's there and that perhaps it's just silent, and not acting, but still being expressed either in other glands or somehow not using the same venom apparatus as the others. But we're not quite sure. We would love to get a genome for this group.
Hello. I know you mentioned the difference between the predatory and defense venoms, and I was wondering if you were going to look more into the chemical makeup of those and whether one or the other would be more useful chemically or like medically.
Yes, yes, there is. The predatory venom we found is the ones that are most useful for biomedical applications. Defensive venom, not so much. So they are quite different in their cocktail biochemical makeup, in the things that they kind of target, the kinds of channels, and how they work.
Hey, I was wondering, so a worm and a fish are two very different organisms with two very different nervous systems.
Right.
I was wondering if you think it would be useful to go after things that go after different kinds of prey, if that makes things more or less likely to do something that's useful in a human.
Can you repeat the last part
So
of the question?
do you think that looking at venom molecules from something that goes after a fish might be, since that's a vertebrate, would that be more likely to give you something useful in a human than looking at something that's injecting worms with venom and might have evolved its venom for those kinds of targets?
Sure, yeah. The things they're targeting are the channels, and for the most part, the channels are conserved in between humans and fish and worms. And a lot of the early drugs that came out are from things that didn't target vertebrates, like, you know, the cone snail that's on the market, they use it for treating, it's a mollusk hunter, and so it can hunt on other mollusks as well.
Yeah, so this is way deeper than.
Yeah, so
okay.
it's not just that because it's a vertebrate, it will be biomedically relevant.
Hi, thank you. This is really very interesting talk on a very interesting field. I was wondering, in the beginning of the talk, you mentioned something about the venom being a cocktail of small molecules, peptides, and then also proteins, and you focus on peptides. Do you understand, or are you working on, or are other people in the field working on, what the synergies between like these cocktails? Are these just mixtures so that something works with the particular target, or is there more to that? Is there some kind of synergy, the small molecule sort of opening the channel or increasing the sensitivity for the peptide or something like that?
Yeah, no, I'm not working on that, but some people have started to look through metabolomics at the small molecules that are found within these venoms and what they can be used for and how they apply. And it's interesting you should mention the mixture of the things. We were just discussing with James, my grad student, the idea of liquid-liquid phase separation. So how the venom enters into the bloodstream and works so quickly has to have some reaction there because they're either in droplets or vesicles or something, and they're flowing through very fast because you see it's instantaneous, right, with the fish. So it's acting both in the peripheral and central nervous system really, really fast. And so how is that happening?
Hello, great talk.
Thank you.
I know you mentioned that venom kind of arose as a result of gene duplication and neofunctionalization. And I know you don't have a genome, but do you think it's possible that there was gene subfunctionalization that actually gave rise to like the predatory versus defensive venom?
That's what I think, but I have no way of, well, we don't know it for sure, but that's what I think, yeah.
Hey, I guess I was curious about how they avoid poisoning themselves. Is it just that it's sequestered, or do they develop actual mutations in their own ion channels? And may that be a way to, you know, select for an ion channel you care about?
Yeah, no, I think it's the way the venom is sequestered. They're actually in these little, we think, vesicles or capsules in how it's delivered. Because there are snails that feed on other snails, right? So they're mollusk hunting snails as well. If you leave them alone in the tank, they can hurt each other and be lethal to each other.
Beautiful talk.
Thank you.
I wondered if you could speak to the types of ion channels. So I noticed from that biochemistry slide that you have such a diversity of peptides. And my first thought in the video is that these would be sodium channels. But then I wonder how many different types of channels do you see? What types that are likely to be targets of this venom?
That's a good question. They target lots of iron channels and receptors. So sodium, potassium, calcium, G-coupled receptors, nicotinic receptors. Again, why they work so well is because this arsenal that's coming at you really is a cluster bomb of biochemical activity. And the mission is to shut down normal physiological functions. So they target a whole range of iron channels and receptors, not just one thing.
Is there somebody right in here?
Oh, couple.
The science is great. Thank you.
Thank you.
I have some sort of simplistic background questions. One, how do you catch them? Two, what are their predators? Three, what is their lifespan? And then four, how do they identify their food?
All right, I'll start with the last one. They identify their food using a siphon. So it's this chemosensory organ that sort of sticks out of the water, because most of the time they're buried under the substrate. And they have this chemosense, like a sniffer, a nose, that will sense if there's prey in the water. And if it is, then they'll put out the proboscis. And then the proboscis, as we saw in the video, can travel really far. And then once they've tethered, they will come up and have a meal. And we capture them either snorkeling or diving. They live in cone snails or in coral reefs. Our terebrids mostly in sandy or muddy bottoms. And we use salad tongs, very sophisticated lab equipment, to grab them very quickly, drop them in a bucket, and then get them back to the lab. And their lifespan, they can live for several years, even in captivity in the lab. As long as you're feeding them, they can live for several years. And was there, I think I missed one. Oh, they're predators. So for the most part, their predators are crabs and lobsters because they don't have soft bodies. So if it has a soft body, then the animal will try to harpoon it. It's why a lot of divers die from trying to collect the cone snails. It's that they see a beautiful shell and then they try to bring it back to the surface, so they put it under their wetsuit, but the animal feels threatened, so it will impale them, and then you're dead. There's no anti-venom for cone snails, because unlike snake bites, which are killing thousands or millions of people around the world and a neglected tropical disease, snail bites are the disease of like the 1%. There are only maybe 24 people or so a year, so it hasn't risen to the level of having an anti-venom being made for them yet.
Hi, great talk. Thank you.
Thank you.
Those videos are really cool, and the snails are pretty sneaky. I'm wondering if there's something else going on there besides just the snails being really slow and sneaking up on the fish, because the fish look almost like really too still or like disoriented. Like especially one of the last videos, it looked like the snail was already almost touching the fish a little bit, and it didn't swim away.
Yeah. It takes a long time to get a shot of the snails hunting fish or worms or even each other. And so these were bottom fish, bottom dwellers, so they didn't swim around too much. But we have videos of them feeding on, I used to show a video of the snails feeding on a clownfish until I went to a kindergarten. And this was before I had kids. And Nemo had just came out, I had no idea. And so we're showing this video of the cone snail eating a clownfish, and the teacher's like, no! Because all the kids are like, no, he's eating Nemo! And I was like, what? I have no idea. But it's just what we have in the aquarium at the time.
My question
So we
was,
don't use clownfish anymore.
my question was, how fast acting is the venom when compared to other venomous animals? Is it just the fact that the animal the snail is hunting is small, or are there properties of the venom that make it fast acting regardless of the scale of the organism it's hunting?
It's fast acting, I think, regardless of the scale of the organism, because when they envenomate humans, it's a matter of minutes, right? So Conus geographus, which is the most lethal species of cone snails, they found it in the Philippines mostly because they were getting caught in the fishing nets, and as the fishermen went in to retrieve the catch, they were being envenomated and just sort of died painless death, because within minutes, to an hour, they were dead, right? And this is a full-grown, like, fisherman kind of person. So they are designed to do what they do quite well. So again, why they're so effective and being repeated all throughout nature is because they work.
Thank you. Great talk. Just curious, are they eating 3 meals a day? Like, how often are they eating, and how bioenergetically expensive is it for them to be producing this venom?
Right. So no, they can usually eat, the fish hunters will eat a fish about their body size. And in the aquarium, if you fed them one a week, that would be it. And then, it takes a while to process, and then they spit out the bones and things, or poop out the bones and things. And so yes, this question of energetics, of the making the venom, because there's so many, molecules being made, it's a good one, and it's being debated, right? Because unlike some snakes where once the snake bites, it has to recover, it takes time, it can't strike again. As you saw in the video, these guys can shoot harpoons right after, rapid fire formation. So, and they're always making the venom, it seems like. So we're not quite sure what the energy cost of making the venom has with it actually being used.
I think we'll take our final question online.
Do you know why so many organisms in Australia are venomous, and why that in particular they are so venomous? Citing the cone snail and the blue ring octopus as examples.
Yeah, I have no idea. God has a special heart for Australia. But it is very much a concentration of venom. And the science that's being done there, they're great people, Glenn King and Ray Norton, and all these guys that are doing fantastic work on venom are Aussies. Yeah, they'll be coming this way, because we have a Gordon conference on venom in August up in Vermont in Mount Snow. And so there'll be a huge contingency of Aussies coming into town if, COVID allows, right? Have to knock wood. Yeah.
All right, well,